There is a country whose modern population exists largely because of a war it never fought. When the American Revolution ended in 1783, and the newly independent United States made clear there was no place in it for those who had sided with the Crown, thousands of British Loyalists fled south to the Bahamas, and they did not travel alone. They brought the people they had enslaved with them. By 1788, just 5 years later, the population of the Bahamas had roughly doubled, reshaped almost overnight by an exodus from a war fought entirely on someone else's soil. More than 2 centuries later, geneticists have finally started reading exactly what that upheaval wrote into the DNA of the people living there today, and the results arrived only weeks before this recording, still fresh enough that the ink on the research is barely dry. The historical outline here is unusually well anchored in documented events. Following Britain's defeat in the American Revolutionary War, Loyalist refugees, families who had backed the losing side, evacuated the newly independent United States in large numbers between 1783 and 1785, resettling across British-held Caribbean territory, with the Bahamas absorbing a substantial share of that migration. Loyalist planters brought enslaved Africans with them, some transported directly from the American mainland, others already part of a longer chain reaching back to the transatlantic slave trade's West African ports of origin. Historical shipping and settlement records for the Bahamas point specifically toward the Gold Coast, present-day Ghana, as a major documented source region for enslaved Africans arriving in the islands during the 18th century, part of the same broader Atlantic system that fed labor into British colonies throughout the Caribbean and North America. Given a demographic transformation this dramatic and this well documented, tied to a specific, dated historical event, and reinforced by written shipping records naming a specific West African source region, you would reasonably expect a population genetic study of the modern Bahamas to eventually confirm the details cleanly. A concentrated source region combined with a datable founding event is close to the ideal setup for exactly this kind of research. Before we look at what researchers actually found, it's worth sitting with just how recent this confirmation is. Unlike most of the populations this channel has profiled, where the genetic study, if it exists at all, was published years or decades ago, the central finding driving this episode was published this same year, meaning we're not describing settled scientific consensus so much as watching a discovery happen in close to real time. In October 2025, a study published in the International Journal of Bahamian Studies, led by Dr. Tanya Sims of the University of the Bahamas and Dr. Michael Pateman of the Bahamas Maritime Museum, working in partnership with Family Tree DNA, analyzed DNA samples from 160 Bahamians. The results showed over 94% of maternal lineages and approximately 73% of paternal lineages tracing to African origins. More specifically, the team identified Y-chromosome markers, including haplogroups E, U175 and EL485, along with mitochondrial haplogroups L2A and L3E, all strongly associated with the Akan ethnolinguistic group centered in present-day Ghana, the same region the historical shipping records had already pointed toward. Here's a question worth sitting with before we go further. If the genetic markers line up this precisely with the documented shipping history, why are the researchers themselves still being cautious about calling it settled? The honest answer is built directly into how the study describes its own conclusions. The research team stated plainly that no exclusive, direct genetic tie to Ghanaians specifically has been fully established, that the markers found are strongly associated with, rather than uniquely limited to, the Akan population, a careful scientific distinction between a compelling, well-supported pattern and airtight proof. That kind of caution is exactly what you want to see in genuinely new research, and it's part of why the team didn't stop at 160 samples. In August 2025, just 2 months before publication, they expanded the project with an additional 45 samples from Cat Island specifically, a location chosen because it was heavily populated during the height of the slave trade, and, crucially, has experienced comparatively little modern migration in and out, making it a strong candidate for preserving an unusually undiluted genetic signal. Early results from that expansion showed high mitochondrial diversity with specific ties to Ghana, reinforcing the broader pattern the first sample set had already suggested. If watching a genetic discovery unfold this close to real time, caveats, expansions, and all, is the kind of thing that pulls you in, that's exactly why it's worth subscribing now. Because this channel exists specifically to catch these stories while they're still developing, rather than waiting years for the tidy, finished version. And if you happen to live somewhere with a documented historical migration event like this one, a specific war, a specific evacuation, a specific ship's manifest, tell us in the comments, because stories with this much paper trail tend to make the strongest episodes. There's an earlier layer of research worth adding here too, because it shows this connection wasn't discovered all at once. A 2011 study by many of the same researchers, published in the American Journal of Physical Anthropology, had already found paternal lineages across 6 different Bahamian island populations, showing African ancestry ranging from just over 71% to nearly 80%, with the specific haplogroup E1b1a M2 predominating across almost every island sampled. But that same study revealed something the 2025 research helps explain more fully: significant island-by-island variation, with Long Island in particular showing an elevated presence of the haplogroup R1b1b1M269, a lineage associated with European ancestry, reflecting the specific, concentrated way Loyalist settlers clustered on certain islands more heavily than others after 1783. The Bahamas, in other words, isn't one uniform genetic population any more than most of the archipelago nations this channel has covered. It's a scattered set of islands, each shaped by exactly where the Loyalist migration happened to land hardest, and where it barely touched at all. It's worth adding some texture to what this Ghana connection actually looks like at the level of individual families, because the broad population percentages can obscure just how specific some of these genetic matches get. Family Tree DNA's own project database, which has been quietly accumulating Bahamian genealogical DNA results for years alongside the formal academic research, shows individual participants matching, marker for marker, with specific ethnic groups across West and Central Africa, some tracing to Ghana directly, others to Togo, Cameroon, Benin, Gambia, Nigeria, and Angola, a spread that reflects the same broader West and Central African source pattern documented in the transatlantic slave trade generally, layered on top of the more concentrated Gold Coast signal the newest research has focused on. One participant's direct paternal line, for instance, matched closely with men from the Bakaka ethnic group of Cameroon, the Baganda of Uganda, and the Mukongo of Angola, a reminder that even a population with a strong documented connection to one specific region rarely turns out to be genetically singular once individual family lines are traced closely enough. There's a further wrinkle worth examining in how this Loyalist migration actually reshaped Bahamian demographics beyond the simple doubling of the population, because the story is more layered than a single wave of arrivals. Historians studying the period note that Loyalist evacuees didn't arrive as one uniform group, making one uniform choice. Some came directly from the American South, particularly Georgia and South Carolina, bringing enslaved people who had themselves already spent one or more generations in North America before the Revolution ever began, meaning their genetic and cultural inheritance by the time they reached the Bahamas already reflected whatever admixture and cultural change had occurred during that earlier American chapter. Others arrived by more circuitous routes, through New York or East Florida, before final resettlement, adding additional layers of movement and time between the original West African departure point and the eventual Bahamian arrival. This matters for how we read the genetic data today, because a population descended from this kind of multi-stage migration carries a more complicated ancestral timeline than a population that crossed the Atlantic once and stayed, even when the underlying West African source region, in this case the Gold Coast, remains broadly consistent across the whole journey. The economic motivations behind the specific demand for Akan and broader Gold Coast labor also deserve a mention, because they mirror a pattern this channel has now documented in several other populations. Gold Coast captives carried a well-established reputation among British and colonial planters for agricultural expertise, discipline, and resistance to certain tropical diseases relative to enslaved people drawn from other West African regions, a reputation that, however dehumanizing its original commercial logic, translated into documented purchasing preferences reflected in shipping records and plantation account books from the period. That targeted preference, driven entirely by the economic interests of enslavers rather than any input from the enslaved themselves, is very likely part of why the genetic signal researchers are now finding reads as concentrated as it does, rather than reflecting the more diffuse, broadly distributed West African ancestry seen in some other Caribbean and American populations, whose founding demand was less regionally specific. For Bahamians today, this research adds a documented genetic layer to a national identity already built visibly around African heritage, most vividly on display every Boxing Day and New Year's Day during Junkanoo, a street festival built on costume, drumming and music with clear West African ceremonial roots, performed today with a scale and civic pride that makes it one of the most visible African diasporic celebrations anywhere in the Caribbean. Dr. Pateman, one of the study's lead researchers, put the connection plainly. Bahamians are now understood to be not only culturally but genetically tied to Ghana, a formal scientific confirmation of something Junkanoo drummers and dancers had, in effect, been performing for generations without needing a laboratory to tell them so. It's worth noting, too, that Junkanoo's own documented history stretches back to the same 18th century period the genetic study is now examining, with some historians tracing its earliest celebrations directly to enslaved communities, marking the few days of freedom granted around Christmas under British colonial custom, meaning the festival and the genome may ultimately turn out to be recording the same population's history through two entirely different mediums, one in rhythm and costume, the other in mitochondrial and Y-chromosome markers. What makes the Bahamas case a genuinely distinct addition to this series is the timing itself. Most of the populations profiled here carry either an old, settled genetic record or an old, unstudied gap. This one is neither. It's an active research program, mid-expansion, adding new islands and new sample sets within the same calendar year this episode is being made, meaning the fullest version of the Bahamas' genetic story doesn't exist yet anywhere. Not in this episode, not in the published literature, not even in the researcher's own current dataset. The American Revolution rewrote this nation's population in a matter of years. It's taken geneticists 2.5 centuries to start reading what that rewriting actually said, and as of right now, they're still only partway through the sentence. Whatever the final chapter of this research turns out to say, it will have been written faster and more publicly than almost any other genetic story this channel has covered, unfolding in real academic time rather than being handed down as a finished conclusion decades after the fact.